Hello children and dear students and dear friends. In continuation with biomolecule quick revision, I am going to discuss about the different questions may be asked in your different exam like CBSE, uh, chemistry exam or CBSE, NET exam or GATE exam or any competitive exams. So you try to know the number of amino acids present in polypeptide chains. You try to remember what is polypeptide, what is polynucleotide, what is polysaccharides. Try to differentiate. Polypeptide make a protein and polynucleotide make a DNA or RNA, and polysaccharide you know that these are carbohydrates. So, number of amino acids present in polypeptide chain. Example, pentapeptide. What is the total number of amino acids? That is 5. Hexapeptide, number of amino acids, that is 6. Heptapeptide, number of amino acids, that is 7. Octapeptide, number of amino acid, that is 8. And decapeptide, that is number of amino acids, that is 10. In this way you can learn and you can complete your question in any competitive exams. But how can you find out the number of peptide bond present in polypeptide chain? You count the number of amino acid minus 1. Suppose it is hexapeptide, then number of amino acid is 6. 6 minus 1, that is 5 peptide bond. In the same way, heptapeptide, the total number of amino acid is 7. So number of peptide bond is 5. In this way question may be asked. Okay. When there is a formation of peptide bond, there is release of one water molecules. So the reaction between acid group of first amino acid and amino group of second amino acid react and make a peptide bond with release of one molecule of water. Next, a polypeptide make a protein. So try to differentiate the word polypeptide or polynucleotide. Again, polynucleotide makes a DNA and RNA. Polysaccharides, you know, these are carbohydrates. Nucleotide, you know, it is the basic unit of nucleic acid. And lot of nucleotide joined together by phosphodiester bond, a bond between two unit of nucleotide and make a polynucleotides. Okay. Next, you try to know amino acid is the basic unit of proteins or polypeptide, and glucose is the basic unit of carbohydrate. It may be oligosaccharides or polysaccharides. Poly means more than 10 unit of glucose. It may be starch, it may be uh, glycogen. Starch generally present in the plant, but glycogen is present in your muscles, in the animals. Next is fatty acid is the basic unit of fat. So you know fatty acid, fat is ester of fatty acid and glycerol. Next is sugar plus base. What type of sugar? It is ribose sugar or deoxyribose sugar. And base. What is base? That is nitrogenous base. It can be adenine, it can be guanine, it can be cytosine, it can be thymine. So if there is a bond present between sugar and base, then this collectively known as nucleoside, not nucleotide. It is nucleoside. Okay. So there is a bond that is glyco sorry there is a N-glycosidic bond, not O-glycosidic. Again, when you add phosphate, then you have to tell It is nucleotide, not nucleoside. Okay, so there is a spelling mistake. Sugar plus base, that is nucleoside, not nucleotide. And next, that is sugar plus base plus phosphate. Again there is spelling mistake. PHOSPHAT, phosphate. Okay, so it is nucleotide. So they, you try to differentiate nucleoside and nucleotide. Okay, and such type of question may be asked that double stranded DNA, single stranded DNA, double stranded DNA, that is ADNA, BDNA, and ZDNA. Okay, so if there is single stranded DNA, in that situation there is no hydrogen bond between the bases. There will be only phosphodiester bond. Okay, and glycosidic bond. If it is double stranded DNA, there must be present of hydrogen bond between two strands of DNA. That's why it is highly stable. That is why it is highly stable. So such type of question may be asked in your competitive exam. In this way, you can revise the chapter, that is biomolecule. That is the syllabus of so many competitive exams. Thank you. I will continue such type of revision in next class. Thank you and have a nice day.